Hi, I'm Matt, and right now I'm going to talk about the Kreb cycle, or the citric acid cycle as it's sometimes called. So basically, as we talked about glycolysis in another video, the end product of that was our 2-pyruvate molecules. There's also other macros that can get you to the Kreb cycle. So through protein breakdown, we could have our amino acids, or through the breakdown of your triglycerides, the fatty acids. But what they all need in order to get into the Kreb cycle is acetyl-CoA. And in this process, through the protein and the carbs, you'll end up losing a carbon molecule in this process, but not from the fats. So basically, like I said, you need your acetyl-CoA to get into the cycle. And the whole purpose of the cycle is oxidation. And what I mean by that is the loss of electrons as we go through this process. So again, we enter through acetyl-CoA. So what happens here is we kind of shave off the CoA and we have our acetyl, which is a 2-carbon structure. It's going to combine with oxaloacetate, which is a 4-carbon molecule. And from there, through the enzyme citrate synthase, we're going to move over to citrate, which is a 6-carbon molecule. So again, we went from 4 plus 2 gave us 6. All right, so here, now through aconitase, we have cis-aconitate and isocitrate. And here in this step, it gets a little interesting because now we lose a carbon molecule in the form of CO2, but we gain an NADH. As we use isocitrate dehydrogenase to go over to the ketoglutate, the same thing happens again. So we lose another carbon through CO2, and we gain another NADH. And so as this process goes on, so if you're keeping track of the carbon molecules, went from 2 to 6, and now we're back down to 4 again. And from here we go around the cycle. We go next to succinyl-CoA to succinate through the enzyme succinyl-CoA synthetase. And at this point we also create a molecule called GTP, which essentially is ATP. It's an easy conversion, and from there, essentially, we just treat it as an ATP. So from succinate, we go up to fumarate through the enzyme succinate dehydrogenase, and in this process, we gain another FADH. From there, we use fumarase to get up to malate, and then malate back to oxaloacetate through the enzyme malate dehydrogenase, which brings us back to the beginning of the cycle. Again, we gain another NADH in this process. So, given the fact that glycolysis gave us 2 pyruvates, we're going to go through the cycle again. So, the overall end product here, we end up with, through each turn in the cycle, we end up with 2 CO2 molecules, 1 ATP in the form of GTP, 3 NADH, and 1 FADH molecule. So, and you can look at each of these two and break them down into ATPs. Each NADH molecule accounts for 2.5 ATPs, and the FADH accounts for 1.5 ATPs. And then from there, we enter the electron transport chain. And I won't talk about that in this video, but in a nutshell, that is pretty much the Krebs cycle.